All right, so we're going to get going here, and before we get into the lecture, we're going to try and use my prop up here for a little tutorial, a really poorly constructed PVC phylogenetic tree that I made this morning and didn't cut the pieces very well, so it kind of falls apart. But I just want to use this to show one thing here, but I'm going to need your help holding it up because the joints don't all together very well. Just hold the bottom too. All right. So here's our evolutionary tree. Right? If we rotate it 90 degrees, that switches between a vertical and a horizontal tree. And what I wanted to show you, I know you can't necessarily perfectly see, that's D, I think that E, C over here, and fashionable B on the bottom. What I wanted to show you was, just since some people were a little confused with this, if you rotate this, we don't need the bottom branch in it. If you rotate this, it doesn't change the meaning of the tree at all. We're just rotating around this node, so that's what I mean when I say you can draw E and D on the left or the right. It hasn't changed the meaning at all. The key thing here is the branching patterns and the groups of organisms in the tree. And so if you imagine a tree being three-dimensional, rather than the way we draw it on a piece of paper, which is two-dimensional, you can imagine these rotations of each of these branches around the tree and whacking the TAs and getting sued and things like that. This is all we do. So I'll leave this in the lab for people to play with. And in the lab, you also will be playing with rotating branches around trees. But the key thing to think about is just, again, the branching order of organisms in the tree, how two organisms sit relative to each other, not how we order them in terms of how we rotated those particular branches when we display them. Okay, so you got to be able to think about that and rotate things in your head, or think about groups of organisms on the tree, rather than just how it's drawn in one setting versus another. All right, now that I have some silicone grease on my hands, and you probably do too, sorry about that. All right, so what we're going to do today is go into a little bit more about character evolution. So what we did previously was look at just trees themselves, the different parts of trees and the groups of taxa on trees, the different ways we can move around taxa and group taxa on trees. And we're going to talk a little bit about that at the beginning today, and then more about mapping characters onto trees, that is, traits of organisms. And then what we're going to do in the next lecture on Wednesday is talk about what we do if we don't actually know the phylogenetic tree. So for everything today and previously, we've assumed we're given a phylogenetic tree. What can we do with it? And what we're going to talk about on Wednesday is what happens if we're not given a phylogenetic tree? How do we figure out what the structure of the tree is supposed to be? So I'm going to go through a little bit more on groupings and trees, and then talk for the rest of the lecture about different aspects of character, traits, and states, some definitions, some detail about how character states change, some terms that we use to refer to patterns of character states on trees, Synapomorphies and Homoplasy, and then right at the end, how we reconstruct the history of character states given a particular tree. So I know we did this right at the end, but I'm going to go over it just again quickly to set you up for a little bit of the discussion of the other aspects of groupings on trees. So we talked about monophyletic groups in the last lecture, that is groups of organisms that share a common ancestor, and when you group them together with all of the descendants of a particular common ancestor, you get a monophyletic group or a clade. And any other grouping of taxa would not be a monophyletic grouping. And I gave you two examples of these non-monophyletic groupings. So the first one was a paraphyletic group. And this is a group of organisms consisting of the common ancestor and some, maybe even most of the descendants of that common ancestor, but not all of them. So I've left out this grouping of E, F, G, and H over here from this group. So this group over here is a paraphyletic group. And then a polyphyletic group is when you have a grouping of organisms that doesn't even include their common ancestor. So flying organisms, like I was saying before, might include the common ancestor of bats and the common ancestor of birds, but not the joint common ancestor of all birds and all bats. And that's a polyphyletic group. Okay, so I'm going to spend a few minutes talking about other aspects of monophyletic or non-monophyletic groups. Everybody up to speed on those? Okay, so we use a lot of terminology, and I want to make sure people are clear when we use this what we mean, although in some cases it's sort of obvious, but I want to do this sort of basic stuff so that in the rest of the class we don't have to go over any of these things again. So we may ask you a question like this, or we may say something that is in essence like this. Are H and G monophyletic? And what we mean by that is, can we draw a monophyletic group that includes H and G and none of the other organisms in question? So you trace to their most recent common ancestor, you draw a grouping around them, and you ask, can you make a monophyletic group out of those taxa to the exclusion of everything else? So the answer for H and G is yes, we can do this for them. Yeah? Right, so I wanted to go over this because we use this terminology and say are H and G monophyletic, and we're implying this process that I'm showing you here. That is, you would look for whether or not you can draw a grouping that would include their common ancestor and no other taxa.
So it's implied that you're including their common ancestor?
It's implied when people say that. That's why I wanted to do this, and we might slip into not saying anything about that, even though formally the monophyletic group should include the common ancestor, and you really should say H and G and their common ancestor. Nobody says that. Okay. So then, you can extend that to saying, are H and F monophyletic? You do the same thing, you find their common ancestor. And for this particular, if we knew that G existed and G was there, H and F are not monophyletic with respect to the other taxa that we're studying here, because we can't draw a grouping around them and exclude everything else. And in many cases, everything is relative to sort of what we know or the group of organisms in question. We're not going to ask you, a question about two new organisms you've never heard of in the context of monophyly. We're going to ask you things about, if we go through a group of organisms and tell you that they're monophyletic, this is what it means. So here's an example of a type of question we might ask, although this is a little bit easier than perhaps we would do in a test. Which of the following in this grouping are not monophyletic? So again, using that abbreviated definition without saying the common ancestor. Yeah, so C, right. So B and C are not monophyletic because we can't make them into a clade and exclude everything else. We would have to exclude A there. We can't do that, so they're not monophyletic. Or the group of B and C is not a monophyletic group. So I'm going to ask you I'll show you another example of a type of question we might ask you. We're going to show you a lot of trees, the evolutionary history of particular groups of organisms, like this one, where we have bacteria, archaea, and the expanded part of the tree for eukaryotes. So look at that tree, remember this tree, and then we might ask you a question like this where we're not showing you the tree. So you're going to have to remember some of the trees that we go through in the class. So based upon that tree, which of the following groups here are not monophyletic? A, right. So if we look at the tree again, you have bacteria and archaea. If we include their common ancestor here, we have eukaryotes that we've excluded as one of the descendants of that common ancestor. We cannot make a clade that includes only bacteria and archaea and not eukaryotes in this tree. So again, this is the type of thing you have to be able to synthesize sort of trees and these phylogenetic terminology in order to be able to answer these types of questions. So what is this pattern called? Right, so it's paraphily because we've excluded one of the descendants of the common ancestor. Okay, so a related term to all of what I've been talking about is relatedness, and you will see this in the book and in some of the lab and probably will be mentioned in parts of the lecture. Someone will say something sort of like with the monophyly, that is that organism X is more closely related to organism Y than it is to organism Z. And when we say that in this class, what we're talking about is in a phylogenetic context. So there are other types of relatedness, like cousins are X% related in humans versus siblings. We're not talking about that here. We're talking about phylogenetic tree relatedness, which is assessed by looking at patterns on the evolutionary tree and looking for where organisms share a common ancestor. And two organisms are more closely related to each other than they are to something else if they share a more recent common ancestor with each other than they do with the other organism. So I'm going to show you this on a tree. So if we ask, is taxon E more closely related to D or to G? The way we answer this is to look at the tree, look at the common ancestors of those groups. So E and G, the common ancestor is here. For E and D, the common ancestor is here. So E and G share a more recent common ancestor, therefore they are more closely related to each other. And again, we will use that terminology throughout. So you have to understand that this is in the context of patterns in the phylogenetic tree and who shares a more recent common ancestor with whom. Yeah?
So it's only chronologically, not necessarily the number of evolutionary steps?
It is not in this, the way we use it is the branching pattern in the tree, not how many changes have occurred along the tree. So that could be a measure of similarity among organisms, how many, because you could have lots of changes happening on one branch to make something look really different. This is just dealing with the historical patterns of shared common ancestry. So it's a sort of a fundamentally different thing. And we will try to not use the same term for that. We might say similarity, degree of similarity, etc., for when we're just looking at how different things are. Was there another? Okay. So then yet another set of terminology that you will see and that we will use a lot is this issue of in-groups versus out-groups. And for most purposes of what we're doing in the class, is a pretty simple way to look at this, which is that in-groups are the group of organisms that we are studying the evolution of, a set of taxa that we are interested in. And then the out-group, relative to the in-group, can be one or more organisms that you know separated out in evolutionary history prior to the existence of the common ancestor of the in-group. That is, they are not members in a cladistic, in a clade, monophyletic clade sense of the in-group. And what this looks like on a tree is this. The out-groups separated out, branched off in this tree prior to the existence of the common ancestor of the in-group. And these are very important for a lot of purposes in studying evolution. We will see a lot of them throughout the class. You've seen some of them in Lab 1 exercises already. So I'll give you an example. If you're studying primate evolution, the human, chimp, gorilla, orangutan thing, you could say those are the in-group. And you could pick an out-group being mouse. Now, you may not know that the mouse lineage separated out prior to the existence of the common ancestor of primates. But the only way that wouldn't be true is that if mice are a derived form of some sort of primate, basically. That is, mice did not evolve from the middle of the primate group. So very frequently we will just state this is an outgroup relative to the other organisms. If there's sort of that logical process, we may have fossil data or other information that would tell us what the outgroup is. we may in fact want to test what is a good outgroup for a particular group. Sometimes we don't know. But the outgroup is best sort of viewed in this phylogenetic sense. Okay. So I think the last or second to last thing related to these groupings of organisms in the tree is something that's very similar to the concept of relatedness. And again, you've seen this in the lab, you'll see this a little bit in the book, and I don't know how much we might drop the term sister this, sister that throughout the lectures, but I just wanted to go through it so that you're ready in case it comes out. I actually do not like this term, but I probably will still use it. So what sister groups are two clades, or one sort of taxon and a major group of organisms, or two just individual taxa that are each other's most closest relatives in the phylogenetic context that you're looking at. In other words, they are the descendants of a single node. So for example, A and B here are the descendants of this node here. They are sister taxa to each other. If we go to this next node here, C is a sister taxon to the group of A and B. And so on. A G and H are sister to each other. F is sister to the group of G and H. And again, we will just sort of let this term slide, and you're expected to sort of understand that it's sort of the next most deeply branching group relative to the organisms that are being talked about. So there's the sort of F versus G and H. Okay, so another practice question before we get into characters. So what is the sister group of this clade D, E, F, G, H? Yeah, ABC, right? So you go back to the last node below the group, and you ask, what is the descendant of that node, the other descendant? And there it is. And so another way of thinking about this is that sister groups sort of originated at the same split event. When a single lineage splits into two, that's when any particular set of sister groups originated relative to each other. Okay, so for all the things I've talked about so far, including the sister taxa relationship, we've assumed that at a particular node, when you have a branch coming into the node, that there are two descendant lineages coming out of that event. Makes sense for many purposes. However, in many cases, we do not want to represent that when we draw an evolutionary tree. Some cases we want to represent something like this, where we have 1, 2, 3, 4, 5 branches coming out of this single node. This is what's called a polytomy. And the main reason we might show polytomies is when we do not know the branching order among the organisms that are shown there. So if we do not know any of the branching order of D, E, F, G, H relative to each other, we will show a polytomy like this. It doesn't necessarily mean that evolution looked like that, although in some cases it might. Evolution can sometimes do these sort of star-like bursts of radiation, but in most cases what we're saying is we just don't know what the relationships are there. And you'll see in the next lecture after today some uses of polytomies. Okay, so that's the wrap-up of everything about sort of groups of organisms and how you think about different patterns of groups of organisms and taxa and trees. Any questions about that before we move on into traits of organisms? Okay. So what I want to do for the rest of the lecture is talk about the traits of organisms and mapping those traits onto a phylogeny for a variety of purposes, onto a phylogenetic tree. So we're going to start out with a little definition here, and haven't actually talked to Brian or Brad about which term they end up using here all the time. I frequently use character trait. When I talk about features of organisms, some people use trait, some people use character. They all mean the same thing. Sort of the formal definition in a lot of evolutionary contexts is a heritable feature of an organism. In some cases, we might use it to refer to just any feature of an organism. It doesn't have to be something that's known to be heritable. So really, the simplest definition is a character is some observable feature of an organism. And again, we might say character or character trait or trait. And then the state for a particular trait is the flavor, the form that that trait takes. And again, we might say character state or state in this case. So an example would be the character could be a heart. If we're comparing different vertebrates, we could say some have a 4-chambered heart, some have a 3-chambered heart, some have no heart at all, some, etc. So in many cases, we might have something like a particular aspect of that character, like 4-chambered and 3-chambered. It is also common to use presence and absence as the character state. So you could also say presence of a heart is a character state for heart, and absence is a character state for heart. And you see this in the lab, I think, for many of the examples there. So what we want to do now is take a phylogenetic approach to looking at characters and asking questions about how character traits and character states map onto a tree. So I'm going to assume we know the evolutionary history here, and we even know what the ancestors' traits and states were like. So I'm going to start with the character being the face, and the state being smiley. They haven't taken Bis002C yet. So we have the common ancestor here of all of these organisms, and they had the smiley face, and then their descendants all inherited the smiley face, and there was no change at all in the character or the character state. So this is what it looks like mapping it onto the tree. That's pretty cluttered. Frequently we only label at the nodes rather than all throughout the branches along there, but you imply that you know what happened along the branches when you label something like this. Sometimes we color the branches to correspond to some table of colors or the things that we show at the tips. Sometimes we throw away the labels on the nodes and just use the colors. These all mean basically the same thing, that this set, all of these taxa today have that character state, and all of the entire group had it from the beginning of their existence. For characters that are inherited from a common ancestor, those are called homologous. Homology is inheritance from a common ancestor. So here, the smiley face in this group is homologous across all the organisms in the group. Okay, that's sort of boring, right? That's just nothing changed for this group of organisms. Now, when you show this, it doesn't mean that other parts of the organisms didn't change. I'm just, in this case, mapping one particular character state onto this tree. Doesn't mean that those organisms are all identical to each other, but for this trait and state, they are. Okay, so let's imagine something changed, and this is, of course, important because there's this, mutations happen, change happens. This is generally called divergent evolution. We're not going to talk about that a lot here, all the nuts and bolts of what's going on inside organisms when features change. Much of that was covered in Bis2B. We're just going to go into, in many cases, focusing on the phenotypes of organisms. Although, in some cases, we may give you some more detailed mechanistic explanations of how something may have changed. So again, let's take the same tree and imagine the common ancestor had the smiley face. But imagine in one of the lineages at some time there was a mutation. like in that lovely bunny video that everybody saw, where eventually, again, we're not showing all of evolution here, eventually this frowny face took over the population here. We've simplified evolution by showing it, and we're just showing the change from one state to another, not all the nuts and bolts of what happened underneath that. But eventually there was a change from this one state to another state, and then all of the descendants after that change inherited what their ancestor had. So this entire clade got the frowny face, and this entire clade got the smiley face. Again, we can throw away most of the labeling there and eventually label it something like this, or even better, label it something like this, where we draw on the tree where the character state change happened. And this tells you a lot of information about the characters and their history in one sort of simple figure. You wouldn't even necessarily have to use the colors here, right? You could just label like you're doing in the lab, make a tick mark on the tree and show where the state change happened. Okay, so two terms that you should have also read about in the lab, but I'm going to go over them here, that are really important in terms of the states that particular characters have. The ancestral state is the state that was present in the common ancestor of a group. So we look at all these organisms, their common ancestor was back here. The common ancestor had the smiley face. So the ancestral state for this group is the smiley face. And then if there's a change in the state in any of the lineages that come out of that common ancestor, they have what's known as a derived state. So the frowny face here is the derived state for that group. And again, I'm sort of going through all these things at a relatively slow pace because we're going to just be launching into and talking about all these things for the rest of the class, and you really have to understand what these mean. So what we're going to do now is talk a little bit about how there are different ways that traits can map onto a tree, in essence. Similar to what I just told you with ancestral and derived traits, but this is a little more about the sort of details of the mapping of traits onto a tree and the history of changes. So the first thing I want to talk about is, I mentioned this previously, so we're interested in monophyletic groups in a lot of cases for evolutionary biology and for general in biology because they share many unique features with each other that are not present in other groups. Why is that? Does anybody know why this is? Yeah.
The chance of the exact same mutation arising in two separate ways.
Yeah, so where would the changes arise that are unique to a particular monophyletic group? Well, they don't arise in the common ancestor. Where, right? So they come to in the branch leading up to the most recent common ancestor. Any changes that happen on this branch have the possibility of being shared among A, B, and C, and absent from D, E, F, and G, and H. So if we have these two monophyletic groups, there are other monophyletic groups here, but if we have these two, changes that occur on this branch may distinguish A, B, and C from the other organisms here. And changes that arise in this branch may also distinguish now D, E, F, G, H from A, B, and C. These shared derived traits are also called synapomorphies. They are shared among a group, and they are a derived state. So it may help people with the terminology. It doesn't necessarily help me because I don't know any Greek or Latin, but here's the roots of the words here. So synapomorphy is derived from Greek. Syn basically means with, that is shared. Apo means away, that is the derived state. And morphe is just the shape or form, that's the trait. So we will use the term synapomorphy a lot, probably. It can be used to refer to a shared derived trait or a shared derived state of a particular trait. And one unfortunate flavor of biology that you're going to have to deal with is that there are exceptions to everything in biology. If we didn't ignore some of those exceptions, we would not be able to talk about much of anything in any context. So there are many groups, many monophyletic groups, that most of the organisms in that group share a derived trait. But perhaps a few of the organisms in that group have lost that trait. So parasitic plants, some of them no longer photosynthesize. But that does not mean that we ignore that 99.9% of plants are photosynthetic. We're going to refer to some of the features of those plants as synapomorphies, even if they're missing from a couple of organisms. I know that the ambiguity is challenging to deal with, but that's just the fact of biology. We still want to call most of those things broadly shared derived traits, even if they're lost by a few members of the group or changed in a few members of the group. Does that synapomorphy make sense to people? Okay, so in a phylogenetic sense, frowny face here is a synapomorphy for this clade. It is the derived state shared among this group. So a shared derived feature. Now, smiley faces might be exciting to me when I'm making my slide presentations, but you are all familiar with lots of shared derived traits in different groups of organisms. So the shared derived traits of mammals, for example, include things that you're familiar with as sort of the way we define mammals in many cases. So mammary glands, the presence of milk, four-chambered heart, hair. Now, subtle details here, maybe the earliest branching thing that some people want to call mammals might not be having all of these features. I study bacteria, so don't stick me on whether or not these are exactly correct. But from looking at groups, and you've heard about different groups of organisms, and sort of many of those shared derived traits of the groups. These synapomorphies are frequently how we then define groups to each other. Those are the features we recognize as being shared among the group. Does that basically make sense? Okay. So I want to now talk about the counter, in essence, to synapomorphy, which is sort of, it's a very important aspect of evolution, and it's a little tricky to think about in some cases. So I'm going to take you sort of through a simple example of this. It's called homoplasy, and the issue is basically summarized in this slide. So if we compare organisms to each other, and we ask for what are their similarities and differences, we can document those similarities and differences between organisms. And as I told you already, if organisms share some feature that came from a common ancestor, that's what we call homology. But there are cases where organisms can share a feature that they separately invented in different branches of the evolutionary tree. So a great example of this is flight in birds and bats. Separate invention of flight in those lineages. The exact mechanisms of flight are not homologous between birds and bats. When you have something like this, where you have a similarity between taxa that is not due to common ancestry, that's generally called homoplasy. And I'm going to show you this in a phylogenetic sense, which is sort of the formal definition of homoplasy. So here's again, I've now made the common ancestor of this group has an indifferent face. So along one lineage, the frowny face evolved, but then along another lineage, separately invented a frowny face in a different lineage in the tree. So if we compared A and B to each other, and to D, E, H, G, and F, they're similar in the sense that they all have frowny faces. But A and B separately invented the frowny face from D, E, F, G, and H. And that separate sort of invention is homoplasy. Those frowny faces are in these different evolutionary events, and therefore they're called homoplasy. or this is an example of homoplasy. The traits are homoplastic traits. And again, if you sort of just label this on the tree, you can see that there was one origin of frowny face here, a separate origin over there. And the bird and bat wings that I just described to you a minute ago is a good example of this. Separate invention of the mechanized flight in these different lineages is an example of homoplasy. I'm not going to go into detail on the different sort of possible explanations for homoplasy. There are lots of different mechanisms that people might throw out to explain the existence of these different similar features in distantly related organisms as separate inventions. One of them you will hear a lot is convergent evolution. We will talk about that a little bit more when we talk about bacterial evolution, but we're just going to leave it as the existence of homoplasy is these separate origins of events without talking about what the sort of underlying mechanism might be behind that. Okay, so that basically makes sense what homoplasy is versus synapomorphy. Synapomorphy, you have a monophyletic group. If there were changes on the branch leading up to that monophyletic group, those are shared derived traits for that group. If you have separate, in different branches in the tree, separate origins of similar features, that is homoplasy. Okay, so what I want to talk about now is the next phase of sort of evolutionary character study, which is what happens, the examples I've shown you are, we assume we know what the ancestral state was. With the smiley face, or even I sort of assumed this with bird and bat flight, etc. What if we don't know what the ancestral state was? What do we do? What we do is use methods called generally character state reconstruction methods to try and come up with a best case scenario for the pattern of traits in the ancestors of organisms. So we have traits in, say, modern organisms. Occasionally you might have traits in past organisms from the fossil record. And we try and infer traits in other parts of the tree using character state reconstruction. We're rarely given what the truth is. We have to figure it out. So let's consider this for the bird and bat flight. Even though there may be many reasons why you think that the common ancestor back here of all mammals and all reptiles and other descendants of these lineages may not have been flying, we're going to do this reconstruction here to see what it's like to figure this out from scratch using this example. So what we do is we throw away all the information about the ancestral character states here. We're going to assume we know nothing about the ancestral character states. We take the states that we know for the modern organisms and we put them at the tip of the tree. And then we test, in theory, you could test every possible pattern of ancestral character states over time in the tree. You could test different either can fly or cannot fly at every node. You could even test for what the change was on different parts of this branch here. We're going to focus on the nodes here and assume that, well, I'll just show you. We're going to focus on reconstructing what happens at the nodes in the tree. So what we ask is basically what is the most plausible pattern for those character states in the ancestors on this tree in every one of these nodes, here, 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 and here. And in theory, they could be anything. So each one of those nodes, in theory, could be either can fly or can't fly. Now, there are many ways where we can simplify the model and throw away some of these possibilities. I'm not going to really show you those right now. We'll talk about that later. What I'm going to do is just posit 3 different scenarios. One of them is the model that I showed you previously with birds and bats having a change in their recent evolution. But let's test out some other models. So here's one. The ancestor here had flight. The ancestor here didn't. The ancestor here didn't. The ancestor here didn't, didn't. That's one possible scenario for the evolution of flight in this group of organisms. Here's another possible pattern. This is the one I showed you with even the ancestor of bats and other mammals not flying, and the ancestor of birds and other dinosaurs or whatever they're related to not flying. And here's a third pattern where the ancestors all could fly in every one of the positions. And then what we're going to do is we're going to count the number of character state changes required given this pattern of ancestral character traits to get the data that we have in the modern organisms. So here's pattern 1, pattern 2, and pattern 3. And we're going to count the number of changes required on those trees. Why are we going to do that? We're going to follow here, at least for now, something called the principle of parsimony, which you've already read about for the lab. And we're going to basically apply the same thing here. The principle of parsimony says that given two competing theories, the simpler one is the one that you should prefer. So if, someone picks up a turtle and puts it down in the front of the room and it's wandering around, and then you see the turtle over there, it's possible that the turtle, went out the door, around, opened the door itself, and then let itself in the other side. A simpler theory would be that it just walked over there. We use parsimony all the time in our daily lives. This is applying that same scenario to an evolutionary question. And what we're going to do is say, we are going to choose the reconstruction scenario that requires the fewest number of evolutionary change events in character states. So we're going to go through all possible character state reconstructions to try and find one that requires the fewest number of changes. That's how we're applying this principle of parsimony to the phylogenetic character state reconstruction. So again, here are the patterns, pattern 1, pattern 2, and pattern 3. If we zoom in on pattern 1, how many state changes are required to match this tree, this data set to those ancestral character states? Quite a few. 1, 2, 3, 4, 5, 6, 7. Now, I am assuming that there are no double changes along particular branches between flight and not flying and then back. It is entirely possible that those have happened in some lineages. But that certainly wouldn't be the simplest model here. We're trying to find the minimum number required for this particular pattern. So we go to tree number pattern 2. How many steps are required? Just 2. Pattern 3, how many steps are required? 6. So which one wins here with the principle of parsimony? Pattern 2. Given these 3 scenarios, pattern 2 is the simpler in the sense of number of character state changes required. And if that all else is equal, we're going to pick that one when we do this character state reconstruction. Does that basically make sense? So I haven't shown you all the possible patterns of character states here. In theory, you could try and reconstruct all possible patterns for this tree. It's not actually that many, but it would take you a little bit of time if you tried to reconstruct all of them. What people frequently do is there are a lot of different tricks you can do in doing this. You can do the 2 scenarios that I did, for example. Force as many of the ancestral character states to be pattern 1 or pattern 2 and see the difference between them. Mostly what we do is we feed them into computer programs that can test lots of the different patterns. We will see in the next lecture for phylogenetic reconstruction that there are some other tricks for doing this when we have a fewer number of organisms in your phylogeny. So we would conclude from this that at least for those things we tested, flight in birds and bats arose separately and therefore is homoplasious. So what we're going to do next is ask, okay, we can reconstruct character states given a tree. And you're going to have to be able to do that. You're going to have to be able to do it in a lot of detail for the next thing that we're going to do. Because we're going to make it a little more complicated. We're going to say, what if we don't know the tree either? What if all we have is these organisms here and their character states? How can we figure out how they're related to each other and the pattern of character states in their ancestors? So what we have to do is not only test different character state reconstructions, we have to test, as they implied in the bunny movie, different phylogenetic trees for organisms. So we're going to compare and contrast multiple phylogenetic trees, each one of them with multiple character state reconstructions. And then we're going to try and find from within that the phylogenetic tree that requires the fewest number of character state changes. So it's going to be this iterative process. Test tree number 1, and then try and find for tree number 1, just like we did here for one tree, what are the fewest changes required for that tree. And then we're going to do tree number 2 and try and find what are the fewest changes for tree number 2. And then tree number 3, what are the fewest changes for tree number 3. And once we've done that for all possible trees, we then compare the trees to each other and ask which of the trees required the fewest number of changes. And that's how we're going to pick the best phylogenetic tree from a selection of many possibilities. So I'm going to just end it right there, and see you on Wednesday.
So